For centuries, Rh-negative blood has been shrouded in mystery and wild theories. Some believed it came from divine bloodlines, others whispered about alien origins. But the real story, the one hidden deep within our DNA, is far more fascinating than any myth. And scientists have finally decoded exactly where it really came from. If you're watching this, there's a good chance you're curious about your own blood type, or perhaps you know someone whose medical records carry that two-letter designation that sets them apart from the majority of humanity. For H-negative. It's a classification that appears simple on the surface, but beneath those words lies a genetic journey spanning millions of years, a tale of mutation and evolution that challenges everything we thought we knew about our species' deep history. The story begins not in a hospital or a laboratory, but in the evolutionary depths of time. To understand where Rh-negative blood truly comes from, we need to travel backward through human history and prehistory, through the development of our species, and even further back to our common ancestors with other primates. The answers we're searching for are written in the language of DNA, a language that scientists have only recently learned to read with precision and clarity. Throughout human civilization, blood has always held tremendous cultural, spiritual, and social significance. Ancient peoples created elaborate systems of meaning around it. They saw blood as the seed of the soul, the carrier of family lineage, the mark of kinship and belonging. When medical science first began categorizing blood types in the early 20th century, it seemed like a breakthrough in understanding human biology. The ABO system was discovered, and later the Rh factor. But as researchers began noticing patterns, an intriguing question emerged. Why did some people lack the Rh antigen entirely? The scientific community initially approached this anomaly with confusion. The absence of a protein that should have been universal seemed like a deficiency, something that didn't fit the neat evolutionary narrative. Some researchers hypothesized that Rh-negative blood was simply a genetic error, a mutation that somehow persisted. Others proposed that it might indicate something fundamentally different about certain population groups. And as the decades passed, the gap between scientific knowledge and public speculation began to widen. This is where the mythology crept in. Internet forums and alternative health websites began spinning increasingly elaborate theories. Some claimed that Rh-negative blood indicated a separate human subspecies, or even extraterrestrial ancestry. These ideas gained traction because the real answer, while not widely publicized, was hiding in academic journals and medical research papers. The truth was far more scientifically elegant than alien theory, but it required understanding molecular genetics at a level that most people had never been exposed to. The real origins of Rh-negative blood lie in a specific genetic mechanism involving something called the RHD gene. This gene is located on chromosome 1, a section of DNA that contains the instructions for building G, the proteins that coat the surface of our red blood cells. When this gene is present and functioning normally, our blood cells produce the Rh antigen, making us Rh positive. But when something happens to this gene, when it gets deleted or damaged, the antigen isn't produced, and a person becomes Rh negative. The fascinating part is understanding exactly how this deletion occurs. Scientists have discovered that the RHD gene sits between two regions called rhesus boxes. These boxes are sections of DNA with nearly identical sequences, like repeated passages in a long book. During the process of genetic recombination, which happens during reproduction when DNA from both parents combines, something remarkable can occur. The cellular machinery sometimes gets confused by these repetitive sequences. It's like a proofreader accidentally aligning two similar passages and cutting out the text between them. When this happens, an unequal crossover event occurs. The DNA machinery performs what's called a recombination, but because the rhesus boxes are oriented in opposite directions and have such similar sequences, the machinery can misalign them. When the crossed strands separate, the result is a complete deletion of the RHD gene from one copy of the chromosome. This individual now carries a haplotype, a genetic combination, that completely lacks the RHD gene. If they inherit this same deletion from both parents, they become Rh negative. This mechanism, this molecular ballet of genetic recombination, is not random in a chaotic sense. Rather, it's predictable at the population level, governed by the laws of probability and the specific structure of our DNA. It happens more frequently in some populations than others, creating the geographic distribution patterns that researchers have observed across different ethnic groups. In Caucasian populations, approximately 41% of people carry at least one copy of the RHD deletion. About 17% are homozygous, meaning they inherited it from both parents, making them Rh negative. In other populations, particularly African and Asian groups, the frequency is much lower, sometimes less than 1%. The reason for these differences lies in human migration and population genetics. After humans began spreading out of Africa tens of thousands of years ago, different populations became somewhat isolated from each other. Within each population, random mutations and genetic variations drifted at different rates. The RHD deletion may have occurred relatively more frequently in ancestral European populations, or perhaps it offered some advantage in certain environments or contexts. Over thousands of generations, what started as a rare mutation became relatively common in some populations, while remaining rare in others. But here's where the story becomes even more profound. The RHD gene deletion isn't actually new to human beings. When scientists studied the genetics of our closest living relatives, primates like chimpanzees and gorillas, they discovered something remarkable. These animals also possess RRH-like genes. They have similar genetic sequences, similar molecular machinery for producing RH antigens. When researchers looked even deeper, examining the genes of other mammals and even bacteria, they found traces of genes related to the Rh system. This discovery fundamentally changed how we understand the origin of Rh-negative blood. It's not a uniquely human phenomenon. It's not something that emerged recently in our species' history. Instead, the Rh factor itself is ancient, dating back millions of years into our shared mammalian evolution. The genetic variation between Rh positive and Rh negative isn't a recent development, but rather an ancient polymorphism, a difference that was already present in our distant hominid ancestors. Consider this. Our evolutionary lineage stretches back approximately 6 million years to our last common ancestor with chimpanzees. Further back, perhaps 25 million years, we share a common ancestor with all apes. And even further back, we share ancestors with all primates, with all mammals, with all vertebrates. The Rh system has been part of this entire story. The genes that code for it have been passed down, modified, and maintained through each generation, through each species, across millions of years of evolution. The RHD gene deletion that creates Rh-negative blood in modern humans represents a variation on this ancient genetic theme. It's not an aberration or mistake. It's simply one of the ways that this ancient system can be expressed. And the fact that this variation exists at significant frequencies in certain human populations tells us something important. Somewhere in the distant past, this deletion must have been maintained at high enough frequencies to persist. Scientists have debated whether the RHD deletion provides any selective advantage. Some have proposed that it might confer resistance to certain diseases, though concrete evidence remains elusive. Others suggest that it simply represents a neutral mutation, one that doesn't significantly harm or help the organism, and therefore can persist at whatever frequency random chance allows. Whatever the case may be, the data clearly shows that Rh-negative blood is not incompatible with human life or reproduction. Millions of people worldwide carry this trait without any significant problems. The inheritance pattern of Rh-negative blood follows a simple Mendelian genetic model. The Rh factor is determined primarily by two genes, RhD and RhCE. Together, these genes provide instructions for making Rh antigen proteins. Because humans have two copies of each gene, one inherited from each parent, the combinations can create different outcomes. If a person inherits the RHD deletion from both parents, they become Rh negative. If they inherit at least one functional RHD gene, they become Rh positive. This straightforward inheritance pattern helps explain why Rh negative blood runs in families. If both of your parents are Rh negative, you will almost certainly be Rh negative. If one parent is Rh negative and the other is Rh positive, there's roughly a 50% chance you'll be Rh negative, depending on the genetic makeup of the Rh positive parent. If both parents are Rh positive but carry the recessive RHD deletion, they can still have Rh negative children, though the probability is lower. The reason Rh negative blood is relatively rare globally, affecting only about 15% of the world's population, has everything to do with this inheritance pattern combined with global population genetics. The deletion is most common in European populations, moderately present in some Middle Eastern and North African populations, and quite rare in East Asian and indigenous populations. As modern genetic science has evolved, researchers have conducted detailed studies across multiple populations to map the exact frequencies and molecular mechanisms of Rh variation. Recent research from 2024 and 2025 has provided unprecedented insight into these mechanisms. A major multi-center study conducted between 2022 and 2025, supported by the Turkish Society of Hematology, examined RHD variants and their molecular characteristics across a diverse population sample. The project, designated as project number 2022-14, represented one of the most comprehensive efforts to understand Rh variant genetics in Turkish populations. The researchers collected samples from multiple university hospital transfusion centers across the country and performed comprehensive genetic analysis using state-of-the-art molecular techniques. They successfully identified the specific mutations and deletions responsible for different RHD phenotypes, cataloging genetic variants that hadn't been previously well characterized in this population. Their work confirmed that while the Rhd deletion is the most common mechanism underlying Rh negative blood, other molecular mechanisms also exist and vary in frequency across populations. The team analyzed approximately 35 samples comprising different Rhd variants, Rhd negative cases, and positive controls, providing a detailed molecular map of Rh variation. Researchers in Saudi Arabia conducted parallel investigations into the molecular basis of Rh blood type variations in their population. Their findings revealed that even within populations that share geographic proximity, there can be different molecular explanations for the same Rh phenotype. Some individuals might be Rh negative due to the classic Rhd deletion, while others might carry different mutations or structural variations in their Rh genes. This molecular diversity helps explain why some Rh negative individuals might have slightly different immune responses or transfusion considerations compared to others. Studies from China and other Asian populations have added further nuance to our understanding. Researchers in Guangzhou conducted detailed analysis of D-elute variants, a special category of Rh negative blood that presents unique challenges in transfusion medicine. They used advanced DNA sequencing technologies, including third-generation single-molecule sequencing, to characterize the exact genetic changes responsible for these variants. Their work, published in 2024, demonstrated that the molecular genetics of Rh blood type extends far beyond the simple binary of positive or negative. There are many gradations and variations, each with its own genetic explanation. The medical significance of understanding Rh blood type genetics became particularly relevant when researchers focused on rare conditions like Rh null blood. These exceptionally rare individuals lack most or all Rh antigens due to different genetic mechanisms than the common Rhd deletion. Finding someone with Rh null blood is extraordinarily difficult, with documented cases numbering in the dozens globally. Recent 2025 research published in the International Immunopharmacology examined the specific genetic causes of Rh null, including novel-based deletions and mutations in Rhce genes, and even genes like Rhag that support Rh protein function. These studies involved pedigree analysis and comprehensive family studies examining multiple generations to trace how these rare mutations segregate through families. The researchers used cutting-edge molecular techniques, including DNA sequencing of all exons and intronic regions of the Rhd gene. These studies revealed that rare blood types often result from multiple layers of genetic variation, with different populations showing different patterns. The complexity increases when you consider that the Rh system isn't just one gene, but rather a cluster of related genes working in concert. The clinical relevance of all this genetic research cannot be overstated. Understanding the molecular basis of Rh blood type variation has profound implications for transfusion medicine and reproductive health. For couples where the mother is Rh negative and the father is Rh positive, there's a risk of hemolytic disease of the newborn if the baby inherits Rh positive blood. This condition, also called Rh incompatibility, can range from mild to severe. In severe cases, it can cause anemia, jaundice, brain damage, or even fetal death. It occurs when the mother's immune system develops antibodies against the Rh antigen on fetal red blood cells, treating the fetal red blood cells as invaders and destroying them. Modern medicine has developed preventive treatments like RhoGAM that can prevent this serious complication. RhoGAM is an injection of antibodies against the Rh antigen that prevents the mother's immune system from recognizing and responding to fetal red blood cells. But the treatment is only possible because we understand the genetic basis of the condition and exactly what causes the incompatibility. For individuals receiving blood transfusions, Rh compatibility is critical. Transfusing Rh positive blood into an Rh negative individual can trigger an immune response, especially if the recipient has previously been sensitized to the Rh antigen. This is why transfusion protocols carefully match donor and recipient blood types. The ongoing research into the molecular varieties of Rh blood type helps blood banks provide safer transfusions by identifying rare variants that might present unique compatibility challenges. The story of Rh negative blood thus interconnects many different aspects of human biology. It touches on molecular genetics, population genetics, evolutionary history, medical practice, and even the cultural narratives we create around biological differences. For much of the 20th century, Rh blood type was simply classified and categorized. People learned whether they were positive or negative and generally didn't think much more about it. But as molecular biology advanced, as DNA sequencing became faster and cheaper, as researchers could examine the actual genetic code rather than just observing the phenotypic outcomes, a much richer understanding emerged. What scientists discovered was that Rh negative blood is not an anomaly or aberration, but rather a natural variation on an ancient genetic system. It's a variation that has been preserved through millions of years of evolution, that exists at significant frequencies in some human populations, and that follows the predictable patterns of Mendelian inheritance. It's neither divine nor alien, but rather the elegant result of how genetic recombination works when it encounters repetitive DNA sequences. The implications of this understanding extend beyond medicine and into our broader perspective on human variation and diversity. For centuries, people have used blood type to construct narratives of difference and otherness. Rh-negative blood, being relatively rare in the global population, sometimes became a focal point for these narratives. But the scientific reality is far more prosaic and in many ways more profound. Rh-negative blood results from a specific molecular mechanism that affects certain populations at different frequencies. It reflects human evolutionary history and population migration patterns. It's a window into how DNA works, how evolution operates, and how natural variation becomes encoded in our genomes. The research conducted between 2024 and 2025 represents the cutting edge of this understanding. Using techniques like high-resolution melting curve analysis, restriction fragment length polymorphism testing, and next-generation sequencing, researchers have moved beyond simply identifying people as Rh positive or Rh negative. They can now identify the specific molecular basis for each individual's Rh blood type. They can trace how different mutations arose and spread through populations. They can predict genetic risks and complications with unprecedented accuracy. For someone who is Rh negative, understanding this science provides a kind of grounding in biological reality. You're not part of some mysterious or special bloodline. You're carrying a genetic variation that your ancestors passed down to you. A variation that emerged through the ordinary processes of mutation and genetic recombination that spread through populations through the ordinary mechanics of inheritance and population genetics. You're part of a population group that has been shaped by human history, migration, and the random drift of genetic variation across millennia. Yet, in another sense, understanding the science makes the reality even more remarkable. The fact that a specific deletion in a gene located on chromosome 1, a deletion that arose through a molecular mistake in genetic recombination, created a variation that has persisted and spread through millions of people across thousands of years. That's an extraordinary story. It shows how complex and intricate human biology is, how the processes that govern life operate at scales we can barely perceive, how our bodies contain evidence of deep evolutionary history. The research published in 2024 from Austria examining novel RHD allele variants and their implications for blood group diagnostics represents another frontier. In this research, clinicians discovered individuals with weakened D expression during routine pre-transfusion testing. These individuals didn't fit neatly into the traditional positive or negative categories. Their genetic analysis revealed novel mutations in the RHD gene that reduced, but didn't eliminate, the production of the D antigen. Understanding these variants has become increasingly important as transfusion medicine becomes more sophisticated and as the potential consequences of blood type incompatibility become better understood. The global distribution of Rh-negative blood tells a story of human migration and population history. In Northern Europe, particularly in Scandinavia and Scotland, Rh-negative blood frequencies can reach 15 to 25% or even higher. As you move south and east in Europe, frequencies generally decrease. In Mediterranean populations, rates are lower. In Africa, Asia, and the Americas, frequencies are typically much lower, often less than 5%. These patterns reflect ancient human migrations and the populations that became somewhat geographically isolated as humans spread across the world. The molecular mechanisms that create these geographic patterns are now becoming clear through research comparing populations. The RHD deletion mechanism that dominates in European populations appears less frequently in Asian populations, where other molecular variants account for Rh-negative blood type. This molecular diversity indicates that Rh-negative blood has arisen multiple times through different genetic mechanisms in different populations. What appears the same, phenotypically, from a clinical perspective, actually has different genetic origins. Understanding this molecular diversity has profound implications for genetic counseling and reproductive medicine. If a couple wants to know the probability of having an Rh-negative child, that probability depends not just on the Rhe, blood types of the parents, but on their specific genetic genotypes, which can only be determined through molecular testing. A person who is serologically Rh-negative might be homozygous for the RHD deletion, or they might have other genetic variations. Knowing the exact genetic basis allows for more precise genetic counseling. The story of Rh-negative blood also illustrates a broader principle in genetics, that the same observable characteristic can result from different underlying genetic causes. This principle, called genetic heterogeneity, is common throughout human genetics. Multiple different mutations in the same gene can produce similar phenotypes. Mutations in different genes can produce the same phenotype. The genetic code is read by protein synthesizing machinery in complex ways, and disruptions at different levels can have similar end results. The study of Rh blood type variation has provided valuable insights into this principle and has helped train a generation of genetic researchers to think complexly about the relationship between genotype and phenotype. As we look toward the future of blood type genetics, several new frontiers are emerging. Researchers are investigating whether certain Rh variants might influence susceptibility to specific diseases. Some preliminary evidence suggests that Rh negative status might provide resistance to certain pathogens or conditions, though this remains an active area of research. The ongoing study of rare blood types like Rh null continues to reveal new genes and genetic mechanisms involved in red blood cell biology. And as more populations have their genetic variants characterized, our understanding of human genetic diversity continues to deepen. The technological revolution in genetic sequencing has been fundamental to this progress. What once required extensive laboratory work and months of analysis can now be done in hours or days. Whole genome sequencing has become affordable enough that entire populations can be studied. This technological revolution has enabled the comparative studies across populations that have revealed the complexity and diversity of Rh blood type genetics. It's also enabled the discovery of rare variants and novel mutations that wouldn't have been identified using older technology. For the general public, the implications of this research are significant but often underappreciated. If you're Rh negative and you're pregnant, this research enables better prenatal care and monitoring. If you need a blood transfusion, this research helps ensure that you receive safe, compatible blood. If you're simply curious about your own genetics and your connection to human evolutionary history, this research provides answers that are grounded in solid science and evidence. The debunking of myths about Rh negative blood represents an important moment in scientific literacy. For decades, fringe theories about divine or extraterrestrial origins circulated freely on the internet and in alternative media. While these theories are entertaining from a storytelling perspective, they distract from the real, scientifically fascinating truth. The actual mechanisms of Rh blood type variation are every bit as remarkable as any fictional origin story, and they're grounded in observable, testable reality. The Rhd gene deletion, the rhesus box sequences, the patterns of genetic recombination, the geographic distribution of variants, the molecular mechanisms that create rare blood types, these are the elements of the real story of Rh negative blood. And this story connects to everything else we know about genetics, evolution, and human biology. Understanding it provides not just answers to questions about blood type, but insights into how life itself is organized and transmitted across generations. The research of the past few years has also highlighted the importance of studying genetic variation across diverse populations. Much of the early genetic research focused primarily on European populations. As researchers have expanded their work to include African, Asian, Middle Eastern, and other populations, they've discovered that genetic variation is far more complex and nuanced than previously understood. The same applies to Rh blood type genetics. By studying multiple populations, researchers have revealed that the mechanisms creating Rh negative blood vary geographically and historically. This recognition of genetic diversity across populations has important ethical implications. It combats essentialist thinking about blood type and other genetic traits. It demonstrates that genetic variation is normal and natural, reflecting human evolutionary history and the adaptive radiation of our species across diverse environments. Understanding this scientific reality helps counteract pseudoscientific racism and essentialism that has sometimes tried to attach grand significance to blood type variation. By placing blood type diversity in its proper context within the framework of population genetics and evolutionary history, we can recognize it for what it really is: a normal, expected outcome of genetic processes operating over time. The modern molecular understanding of Rh blood type thus represents a triumph of scientific methodology. Through careful observation, hypothesis formation, experimentation, and refinement of theories based on evidence, scientists have moved from mystification to understanding. They've identified the specific genetic mechanism responsible for the most common form of Rh negative blood. They've traced its evolutionary origins millions of years into the past. They've mapped its distribution across human populations and explained the patterns through population genetics. They've discovered that the mechanism isn't universal, but varies across populations. And they continue to refine and deepen this understanding through ongoing research. In conclusion, the answer to the question of where Rh negative blood comes from is remarkably clear from a scientific perspective. It comes from a deletion in the Rhd gene that arose through genetic recombination involving rhesus box sequences on chromosome 1. This deletion represents a variation on an ancient genetic system that has been present in mammals for millions of years. It became relatively common in some human populations through the processes of genetic drift and population history. It's inherited according to simple Mendelian principles, and it can now be identified and characterized at the molecular level using modern genetic technologies. This scientific understanding didn't emerge all at once, but rather accumulated gradually through decades of research. The mysteries and uncertainties that once surrounded Rh-negative blood have been progressively resolved through improved technology, more sophisticated experimental designs, and increasingly detailed knowledge of molecular genetics. The research of 2024 and 2025 represents the latest chapter in this ongoing story, with studies across multiple populations revealing the molecular diversity of Rh blood type genetics. For anyone carrying Rh-negative blood in their veins, the knowledge that this trait has ancient evolutionary roots, that it reflects human history and population genetics, that it's now fully understood at the molecular level, provides a kind of grounding. You're not carrying a mark of divine favor or extraterrestrial ancestry. You're carrying a genetic variation that is part of your human heritage, one that connects you to the deep evolutionary history of our species and to the millions of other people around the world who share this trait. That's the true story of Rh-negative blood, and it's far more interesting than any myth.